So you can upload your microarray or RNA-seq data and look at the different pathways, and in a convenient way come up with a picture of how things look across the different diseases, infectious diseases you're looking at. And then perform a network analysis of several types. And okay, so typically you start by uploading your gene data, and you can visualize it with an integrated kind of network browser which loads into Cerebral. You can do pathway overrepresentation. So, for example, if you're interested in any of these pathways, how many genes in those pathways have been affected by whatever treatment you loaded here? What are the chances of that happening? And then here we show the follow-up with biochemical tests. And the same thing with transcription factor binding sites to quickly come up with what you're interested in. What the data shows is which transcription factors are likely involved in your response. And then map it onto a network. So for this example, it's just showing gene expression in response to IDR peptides. Looks like there's a large involvement of type 1 interferon. My own work is more not user-friendly. It's more based on publicly available code. If you've used our code, Bioconductor, you know that there's a steep learning curve. It looks almost like a cliff. So in order to kind of support the wet lab microarray work, I added a little web front end with Django. If you're a web developer, you know about Django. There's thousands of commercial sites using it now. It provides things like automatic persistence of your data to databases, etc. We've got probably around 100 public experiments in the repository now. And the idea is you just load lots and lots of data. You can calculate differential expression on all the major platforms, from spotted microarrays, AFI, beta arrays from Illumina, RNA-seq. And then in a batch process, you just start the process running, and it does the overrepresentation analysis, clustering analysis of the interactome. And then the idea is it would lead to some kind of identification of something that's relevant to the disease of interest. So I'm particularly interested in inflammatory diseases, specifically inflammatory bowel disease is what my fellowship is funding. So loading just a set of about a dozen geo experiments, finding what's important, what's not, made some headway into identifying what are the key regulators, what are the key genes involved in turning on the anti-inflammatory action of commensals that's not seen in pathogens. So, kind of in summary, there's been I'll just let you read through these. Lots of presentations and papers have been done on this work, and other major outcomes are patents filed for peptide therapeutics, as well as licensing and preclinical development, and new collaborations have been formed or ongoing. In terms of future directions, again, publications, IDR as a host-directed therapy, and commercialization. Continuing support for an ATB is turning out to be problematic, but one of the head people, David Lynn, has now he's a PI now in centered Ireland, so he's got some people as well. In terms of just to end on the meta-analysis front, happily I've got a fellowship, so I can keep going on public data with MetaGex. And some of the key areas that we'll be looking at is, again, commensal pathogenic bacteria, sepsis, and how to properly model it in animals or in vitro blood work, and then cystic fibrosis gene linked disease. And here's just a quick picture of some of the people involved. And there.
Questions for Chris? What we're going to do is, if you just kind of put your hand up a little bit, we're going to try to bring a microphone to you so that everybody can hear your question clearly.